Good morning, students. Today we will take up a new topic from the same chapter. Today we will study about the experimental proof of a DNA replication. Replication is the formation of exact carbon copies of a substance, and it occurs in case of DNA, and it takes place during S phase of cell cycle. So in this video lesson we will study both the experiment to prove the semi-conservative mode of replication of DNA. Now, DNA replication, the unique process of making an identical copy of a double-stranded DNA using existing DNA as a template for the synthesis of new strand is called DNA replication. So, replication is a biological polymerization process which proceeds in a sequential manner. Okay, that is initiation, then elongation, then termination. And the whole process takes place with the help of enzyme where DNA dependent DNA polymerase being the chief enzyme. Okay, so in 1953, Watson and Crick proposed the mechanism of replication of DNA. And here you can see their original statement regarding replication of DNA is as follows. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Okay. So we will see what is the semi-conservative replication of DNA. Semi-conservative replication of DNA means in this one half of the parent structure passes into each replica while the second half is built anew. Okay. So Watson and Crick suggested that the two strands of DNA molecule uncoil and separate, and each strand serve as a template for the synthesis of a new complementary alongside. The template and its complement then form a new DNA double strand identical to the original DNA molecule. And the sequence of bases which should be present in the new strand can be easily predicted because these would be complementary to the base present in the old strand. Means A will pair with T, T with A, C with G, or G with C. Thus, the two daughter DNA molecules are formed from the parent DNA molecule, and these are identical to the parent molecule. So each daughter DNA molecule consists of one old, that is the parental strand, and one new strand, since only one parent strand is conserved in each daughter molecule. So this mode of replication is said to be semi-conservative. Okay, so here you can see that the two strands of the DNA molecule unwind and separate from each other. Okay, in a zipper-like fashion, and it occurs due to the breakage of the weak bonds between the base pairs. Okay, now each strand here act as a template for the synthesis of this one new strand. Here you can see this one is the new strand, the red one. Now the bases in the strand, it acts as a template and it decides the complementary bases to be present in the new strand. Okay, so the result is the formation of the two daughter DNA, okay, which are identical to each other and to the parent molecule. So each daughter molecule has one strand, okay, from the parent, and one is new. Now in other words, during replication, half of the original DNA molecule is conserved and the complementary half is newly synthesized by base pairing, and this is called semi-conservative mode of replication of DNA. Now, how DNA is replicated? There are three hypotheses. Okay. Now, these three hypotheses are: first one is conservative replication, that is, replicated DNA result in one old double strand DNA and one new double strands of DNA. Here you can see this is the conservative replication. This is the parental DNA strand. Then one daughter DNA molecule is all new and the other one is old one. Next is dispersive, where the replicated DNA result in each strand double strands with segments of old and new DNA. Okay, that means it contains a mixture of old and new DNA. And then semi-conservative replication, that is, each double strand has one old strand and one new strand. Means each daughter DNA molecule contains one new strand and one old strand. Okay. Now, what are the experimental proof? That is, what are the experiment to prove the semi-conservative replication of DNA? So first we will see the first experiment. Okay. In 1958, J.H. Taylor et al. Demonstrated the semi-conservative mode of replication in DNA and chromosomes in root tip of Vishia faba. After the incorporation of radioactive thymidine, the root tips were transferred to unlabeled medium. Okay, and the radioactive chromosome with labeled medium having this radioactive thymidine appeared. Okay, now after the replication of DNA and constitution of chromosome, following results were obtained. Here, please make it a correction. Here it is written following results were obtained. Okay, so here you can see what was the experiment done. So Taylor et al. Supplied roots of Vishia faba with radioactive tritiated thymidine for a few days till the chromosome of dividing cell becomes radioactive. Now, thereafter, the radioactive chemical was withdrawn and replaced with non-radioactive chemical. Every cell cycle was studied and it was found that in first generation, after removing tritiated thymidine, one of the two chromatid of each chromosome remained radioactive while the second chromatid was non-radioactive. And in the second cell generation, 50%, here you can see 50% of the chromosome were unlabeled, while in the remaining 50% labeling remained only in one of the two strands. So it proved that one strand of the chromosome persists in daughter while the second one is formed anew at the time of each replication. Okay, so these were the result. It was found in first generation that radioactivity was uniformly distributed in both the chromosome. In such cases, original strand of DNA double helix was labeled with radioactive thymidine and newly formed strand was non-labeled. Here you can see labeled thymidine and then the newly one is non-labeled. Now during the second generation, only one of the two chromosome represented radioactivity by showing one strand radioactive and the other one as non-radioactive, that is the newly formed. Okay, so this proves the semi-conservative replication of chromosome. Okay, now next is DNA replication, how it is semi-conservative. Next experiment was conducted by Michelson and Stahl in 1958. Okay, so Michelson and Stahl's experiment was conducted to explain the semi-conservative mode of replication. Okay, it was conducted in 1958 and they carried out series of experiment in bacteria E. coli. Okay, what they did, they grew the bacterium E. coli for many generations in a medium containing heavy isotope of nitrogen N15. Okay, now heavy and normal are the two isotopes of nitrogen which can be distinguished based on their densities by centrifugation in cesium chloride. Okay. Then the medium contained ammonium chloride as only the source of nitrogen, and the DNA of the bacterial cell was labeled with N15. Okay. That means they cultured E. coli in the medium constituting ammonium chloride over many generations, and as a result, N15 was integrated into the bacterial cell. Okay. Now, later what they did? Later they revised ammonium chloride medium to normal ammonium chloride, that is with N15 to N14. Okay. Now, the bacterial cell contain N15 and which were transferred to the medium containing normal nitrogen, that is N14. Okay. Now at a regular interval of time they took the sample and checked for the density of the DNA. Okay. So this is the density gradient centrifugation. Okay. And this tested the presence of heavy DNA and normal DNA. Okay, so for this the isolated DNA was added to tubes having cesium chloride, and the tubes were provided with the centrifugal force of 50,000 revolution per minute for many hours till DNA came to lie at a fixed position in the tube. Okay. Now, comparing the heaviness of the DNA of each generation, it was found that the DNA of the generation having completely heavy isotope was the heaviest. Okay, and based on these, then the result obtained and the interpretation was done. Okay, now centrifugation is based on the fact that any object, that is the separation of the particle, occur into zone on the basis of their density differences. Here you can see that is each particle with sediment only up to the position in centrifuge tube at which the gradient density is equal to its own density. Okay. So here first the DNA is mixed with cesium chloride, then it is centrifuged at a very high speed. Okay, for many hours. As the tube spins, the opposing forces of sedimentation and diffuses produces a stable gradient of cesium chloride with the lightest density at the top and the heaviest at the bottom. And as the cesium chloride gradient is formed, DNA comes to at equilibrium in gradient where its density is equal to the density of the surrounding cesium chloride. Okay. Now this is the figure, okay, of the separation of the DNA by centrifugation. So at specific, after the bacterial cell containing N15 were transferred to the medium containing normal nitrogen, then after specific times corresponding to replication time of DNA, samples were taken and DNA was isolated by high speed and evaluated for its density. Okay. So after that, what it was done, first it was seen, that means after 20 minutes the density of DNA was intermediate between the heavier N15 DNA and the lighter N14 DNA. Okay. So it was possible only when DNA contained equal amounts of N15 and N14. And this is exactly what happens when each strand acts as a template and uses the new material to make another strand during replication. Okay. Now here N15, here you can see this one, the red one, the N15 acted as a template and made new complementary N14 strand from the raw material available. So each new DNA molecule had one N15 and one N14 strand, hence had the density midway between N15 and N14. Okay. Now after the second replication, that is after 40 minutes, the DNA extracted from the culture showed that the density of one half of DNA was between N14 and N15, identical to the one obtained during first replication, and the other half was in N14 density region. Okay, this is N14, this is N14, and this is the intermediate, that means the light and the hybrid. Okay, so as it did not contain any new heavy nitrogen or N15, it confirmed the semi-conservative mode of replication. So DNA extracted from the culture contained equal amount of hybrid DNA and light DNA. So what was the observation? That the sample had bacterial DNA with an intermediate density, and after 40 minutes the sample contained DNA with both intermediate and light densities in the same proportion. So what was the conclusion drawn? So based on the observation and experimental result, Michelson and Stahl concluded that DNA molecule can replicate in a semi-conservative manner. Okay, now let's study this one. Comparing the heaviness of the DNA of each generation, it was found that DNA of the generation having completely heavy isotope was the heaviest. So in the first generation, after bringing the bacteria on culture having normal nitrogen, DNA settled at a slightly higher level, indicating that it has become lighter as compared to the DNA of its parent generation. Okay, and then in the second generation, after 40 minutes, okay, here you can see DNA formed two sediments, one at the level of DNA of first generation, okay, and the second at still higher level. So it is clear that DNA of the first generation is intermediate and it contains both N15 and N14. And then in the second generation there were two types of DNA, one of the intermediate heaviness having N15 and N14, and second fraction of lighter DNA having N14 and N14. Okay, I hope you are getting it, that in the first generation, okay, when the DNA was isolated after first generation, it produces a single band only. Okay, so this band, however, this band was higher, intermediate in density between the heavy N15 and light N14. This indicates that DNA molecule made in first round of replication was a hybrid of light and heavy DNA. Okay, and this fit with the dispersive and semi-conservative model but not with the conservative model. Okay, then in generation 2, information from this determines which model is fit, that is it may be semi-conservative or it may be dispersive. Okay, so when second generation here DNA was centrifuged, it produces two band. One was in the same proportion as the intermediate band from the first generation. Okay, while the second one was the a little higher one. Okay, so this result that DNA was being replicated in semi-conservative manner. Okay, and in the same way, okay, generation 3 and generation 4. So if it was semi-conservative model, each hybrid DNA molecule formed from second generation would be expected to give rise to a hybrid molecule and a light molecule in third generation. Okay, so thus over third generation and fourth generation, it is expected that hybrid band, here you can see, would become progressively fainter, that is it represents a smaller fraction of total DNA, and light band to become stronger, that it represents a longer fraction, here you can see. Okay, from the percentage here you can see that the ratio of the two types of DNA first was 50-50. Okay, in the second generation, then in the third generation after 60 minutes again there were two types of DNA, intermediate and then light. However, the percentage of intermediate, this one was reduced from 50 to 25%, while that of the lighter fraction increased from 50 to 75%. Then again in the fourth generation after 80 minutes the percentage of intermediate fraction was decreased further to 12% or 12.5%, while that of the lighter fraction increased from 75% to 87%. So all this could happen when in each generation out of the two strands of DNA, one was obtained from the parent double strand while the second was formed fresh from the nucleotide. Okay, so this proves semi-conservative nature of DNA replication. Okay, I hope this is clear. Now, let's compare all these three hypotheses. Okay, now this is the conservative replication. Okay, where in the first replication, here you see original and one is the only newly formed. Then in dispersive there are the patches of both, and in semi conservative one is new and the other one is the parental one. So dispersive predicts that each replicated DNA molecule should be a patchwork of parental DNA and daughter DNA. That is, every round of replication will produce both heavy and lighter section. Okay, and in conservative replication, after one round of replication, the original heavy DNA along with one new light DNA, and if they went through second replication, then there would be one heavy DNA and then there will be 3 light DNA. Okay, so this indicates that the heavy DNA never disappears completely in this conservative mode of replication, where we have seen in semi conservative, okay, that the percentage of the intermediate fraction was decreased and the lighter fraction was being increased. Okay, so and it never produces the hybrid molecules also in the conservative replication. Okay, so from this it proves that the nature of the DNA replication is semi conservative. Okay, so I hope this is clear to you. So that's all for today. Next we will deal with the mechanism of DNA replication. Okay, that's all. Thank you.